And what we're going to look at now is how we can use titrations for working out the concentration of a solution that we don't know about. Right? Working out the concentration of an unknown solution, as it's called. Oh, that's just waffle. So, for example, imagine we went down to the shop and we bought ourselves a bottle of fancy vinegar, and on the label the fancy vinegar said that it had a certain percentage of acetic acid in it, or fudic acid as it's sometimes called, and we wanted to check that. How do we do an experiment to measure the concentration of acetic acid or ethanoic acid in the vinegar? We can do it by going to our knowledge of the sorts of chemical reactions that ethanoic acid can do, that we've just been talking about. Right? So what we're going to do is we're going to start by taking an accurately known volume of our vinegar. Right? So let's say we prepare 20 ml of our vinegar out of the bottle, and we put it in the little flask of our titration. We put it in the bottom, in the little flask, right? And then what we're going to do is in the burette, we're going to put a solution of sodium hydroxide. And this is a solution of sodium hydroxide that we accurately know the concentration of. And so we very carefully prepared that solution to be a particular concentration, or we've made it and then checked its concentration somehow afterwards, but we accurately know the concentration of the sodium hydroxide solution. And so we're going to call that a standard solution. And I think you made one of these actually in the first lab, yeah, with sodium carbonate or something, where you weighed it out accurately and then you put it in a volumetric flask, and so then you accurately knew the concentration. Right? So the point is, we don't know the concentration of the vinegar solution, but we accurately know the concentration of the sodium hydroxide solution. Okay, and so here's just a little picture showing the situation. So in the burette we have our sodium hydroxide that we accurately know the concentration of, and in the little flask we have our vinegar or our ethanoic acid solution, which we don't know the concentration of, but we do know the volume of. Okay, so what we're going to do then is we're going to carry out the titration, and we could do it like we did in the lab yesterday, right? We put a pH meter in our little flask, and we start slowly adding our sodium hydroxide, and we follow how the pH changes. And we're going to get a curve that looks like pretty much what you got yesterday, because the thing that's in the vinegar that reacts with the hydroxide is the ethanoic acid. Once we've got this curve, then the key thing that we want to go looking for is the volume of sodium hydroxide that we had to add to get to the equivalence point. Right? So we add hydroxide and the curve goes up, and then once we're done, we can go back and we can look at where point S is, halfway up the steep part, and we can look and see what volume of sodium hydroxide we had to add to get to the equivalence point. Now, in order to measure this volume very accurately, I don't know whether the guys talked to you yesterday about this or not with the burettes, but you should be able to read a burette to 2 decimal places. Right? Did they say this? You didn't really have to read them yesterday, but you are going to have to today, and in the other labs where we do titrations. So here's a picture of what it looks like, and you may have noticed that when you try and look at the top of the solution in your burette, in fact, it's flat across the top, but then it's curved at the bottom. This is something called the meniscus, and it's a consequence of water having surface tension, the way that it does. You know about water beading on your car and on slippery things because of the surface tension. When you put it in a tube, it ends up looking like this, and when you read the burettes are set up for you to read the bottom of the meniscus. Right? So not the top flat part, but the bottom of the curvy bit. And so if that was your burette, there's 25 at the top and there's 25.5 down the bottom. That would be 25.30. Obviously this is horribly magnified, but you get the point. Whereas if it's between those things, it's 25.35. So you should be able to at least read them that accurately. Sometimes people, well, even I sometimes will get excited and suggest that if it's on the line, then it's 0.30. If it's halfway, it's 0.35. If you can tell that it's past the line but not quite halfway, then you call it like 0.32. And if it's past halfway but not quite to the next one, you call it 0.38. You can do that as well if you want. But certainly 2 decimal places. The burettes are made to be read that accurately, so you should read them that accurately. Right? But you'll talk about this more this afternoon. Okay, so now the key thing to remember, of course, is that the equivalence point in your titration is the point where you've added just the right amount of hydroxide to react with all of the acid molecules that were originally present in your little flask. All right, that's, if you like, the definition of the equivalence point. The point in the titration where we've added enough hydroxide to react with all of the acid molecules. And here's the chemical reaction, yet again, that goes on when you add the hydroxide to the ethanoic acid. And so if we do the titration, and then we can go back and we can look at where the equivalence point was, and we can measure what volume of sodium hydroxide we had to add to get to that equivalence point, then, because we know the concentration of the sodium hydroxide solution as well, we can work out the amount of hydroxide ions that we needed to get to the equivalence point. And so let's imagine that we had to add 18.5 mils of the sodium hydroxide solution to get to the equivalence point. And let's further imagine that the concentration of that solution you knew accurately to be 0.012 moles per litre. Right? So you've worked that out independently somehow. Then the amount of hydroxide that you had to add to get to the equivalence point, we can work out because we know that the amount is equal to the concentration times the volume. So it's 0.012 moles per litre times 0.01850 litres. And that comes out to be 2.22 times 10 to the negative 4 moles. We happy? So this is the amount. Are you happy because you're not happy, or happy because you're not finished? Okay, that's fine. Do you imagine you're going to be happy when you are, though? So all we're doing is working out the amount of hydroxide we've had to add to get to the equivalence point. And of course the point is that because this equation has a one-to-one relationship between the amount of acetic acid that reacts and the amount of hydroxide that reacts, if we know the amount of hydroxide that we need to add to get to the equivalence point, that must be the same as the amount of ethanoic acid molecules that were there in the first place. Yeah? So if we needed to add 2.22 times 10 to the negative 4 moles of hydroxide ions to reach the equivalence point, then the vinegar solution that we started with in our little flask must have contained 2.22 times 10 to the negative 4 moles of ethanoic acid molecules. Right? Must be the same amount because of that one-to-one ratio between those two reactants. And so the titration then enables us to work out the amount of ethanoic acid that was in our sample of vinegar. And then because we accurately measured out the volume of that vinegar solution, that enables us to work out the concentration of the ethanoic acid in the vinegar solution. It's just the amount divided by the volume. And so the concentration turns out to be 1.11 times 10 to the negative 2 moles per litre. And that's what the answer to the question posed is. But of course we could then get you to work out what the pH of that vinegar solution was. And so that's then going back to a different sort of calculation like we did yesterday, where now that you know what the concentration of the weak acid is, you could work out the pH. Yeah? So we could tie both of those different sorts of things together if we were feeling mean. Now the last little bit is just to point out that normally when you do these sorts of things, you don't do them with a pH meter sitting in the solution, right? If you do it with a pH meter, then you have to do the entire titration, get the whole curve, then go back and find the equivalence point and do the calculation from there. But normally we don't use a pH meter. But if we don't, we still need to know accurately where the equivalence point is. We need to know how much hydroxide we have to add to get to the equivalence point. And so the way that we do that is by using something called an indicator. Right? And you played with an indicator yesterday a little bit, that pink stuff. The indicator is a little molecule that you stick in to your little flask when you do the titration that changes colour when you reach the equivalence point. So if you like, it indicates the equivalence point in the titration. So the indicator itself is normally a weak acid, and when it donates a proton, it changes colour, right? So for example, I think the one you used yesterday was phenolphthalein, is that right? Yeah. So that's what a phenolphthalein molecule looks like. Please don't copy that down, it's completely not important. And if your solution is acidic, if the pH is low, then this is the way that the phenolphthalein is, and so it has that structure which is completely unimportant for you. But when the solution becomes basic, oh, it doesn't do that, it doesn't disappear, it goes that way, right? It goes pink, and as you saw, it kind of is almost that pink colour. And you can see, hopefully, that at the top there's a couple of OHs, so there's an oxygen with a hydrogen on it, and over here those hydrogens have gone, right? So this is the acid form, and then it donates those hydrogens away and it turns into that. And there's all sorts of consequences for the bonds, the way that the bonds work and all these rings and stuff. And the result is that it goes this bright pink colour, right? So that indicates to you when you've reached the equivalence point in an ideal world. The pH at which the indicator changes... No, I said don't write this down, I'm just going to read it. The pH at which the indicator changes colour varies from indicator to indicator. So different indicators have different pHs at which they change colour. So phenolphthalein changes colour at a particular pH. Bromothymol blue is different. Thymol green is different. To be suitable for our titration, what we want is the pH at which the indicator changes colour to be about the same as the pH of the equivalence point. Right? That kind of makes sense. And so what you can do is you can look up in a book. Go away. Go away. What are called pKin values. Right? And so because they're a weak acid, then they have a Ka as well, just like any other acid. But because we call these indicators, then there's another way of talking about the K. So pKin is just the pH at which, well, it gives you an idea of what pH the solution is when the thing changes colour, right? I'm getting to the important point. So you can look up these pKin values, and what you want is for the pK of the indicator to be about the same as the pH of the equivalence point for your titration. Right? So you kind of have a feel for what the pH of the titration of the equivalence point is going to be because you know what sort of titration curve you're talking about. So if it's a strong acid, strong base, the equivalence point pH is 7. If it's a weak acid, strong base, it's bigger than 7. It's like 8, 8.5 or something. And so you pick an indicator whose pKin is about the same as the pH of the equivalence point. Right? And so you go to a book, and here's a picture of what you get in a book of all these different acid-base indicators. And over here, you have these pK values. This is actually called pKa, not pKin, but it's the same thing. And so what you want is for that number there to match up with what you expect the pH of your equivalence point is going to be. And then you just put a few drops of that in, and then you do the titration, and when it changes colour, that tells you when you've reached the equivalence point. And so then you can do your calculations.